we could all take him as an example of what it is to get the job done. Justin, your talk, you push the magic button, is titled Detecting the Source, creating a novel tool to detect disease-associated gene expression in the retina. Justin.
Okay, thanks. So today I'll be specifically talking about a novel tool that I created to detect the expression of a particular molecule that might cause blindness in age-related macular degeneration. So to begin, I think I'll give you a bit of background about age-related macular degeneration, or AMD as I'll refer to it from now on. So this is the leading cause of vision loss in the elderly in industrialized countries. It affects about 1 in 20 people over the age of 60. And there's no available treatment right now for the early stages of this disease. So overall, the causes of vision loss in this condition remain mostly elusive. For about 90% of people who suffer from this condition, they have a particular type of AMD called dry AMD. And this condition is characterized by the accumulation of what are called drusen in the retina. So this is an image here, looking back in someone's eye, as if you were looking through my pupil. And this is the retina, so this is the optic nerve here. And these little yellow deposits that you see around the center, a region called the macula, an area necessary for high acuity or 20/20 type of vision, are these deposits. And those are drusen deposits that I'm talking about. So they're extracellular deposits just beneath photoreceptors, rods, and cones of the eye that are necessary for us to see. So importantly, these drusen deposits contain various disease-associated components found in other diseases. One such component is termed the amyloid beta protein. And this is a protein characterized for its involvement in Alzheimer's disease. It accumulates in the plaques of Alzheimer's disease patients' brains around memory-associated structures where it causes cell death and memory loss. So recently we've been finding this molecule in these drusen deposits around photoreceptors of the eye. And its data has recently accumulated to suggest that this molecule might be involved in vision loss in AMD. So a group that I've been working with has wanted to detect this molecule in a rhesus macaque model of the disease, which is highly representative of the human condition, so that we can hopefully develop treatments at some point for people that suffer from this disease. So before I continue on, I think I need to go over how the A-beta molecule is produced. So like any protein, information for making that protein is encoded in genes, or DNA. This DNA, this genetic information, is then transcribed into a messenger molecule termed messenger RNA. And the particular gene that does this is called amyloid precursor mRNA, APP mRNA, as I'll refer to it. APP mRNA travels to other parts of cells where it's then translated into proteins. And proteins are the functional units of cells. They move around and allow cells to do their work. In disease states, however, proteins often cause cell abnormalities and can lead to cell death. So we've been interested in detecting this molecule in a monkey model of the human disease AMD, and the retina specifically. So we want to see where this stuff is made and how it might cause vision loss in these animals, and how representative that is of the human condition. So a technique that we've used to detect the A-beta protein in the retina is termed immunohistochemistry. It sounds like a complicated term, but when you break it down, it really isn't that complicated. So immuno indicates the use of the immune system. We use antibodies that bind to proteins like suction cups in tissue. Histo refers to histology, or the study of tissue, and chemistry indicates a chemical bond. So in this technique, we add little fluorescent labels to antibodies, and then we watch these antibodies over a section of, say, retinal tissue, and if a certain disease component is expressed somewhere in that brain tissue or retinal tissue, then these antibodies stick to it, and you can see those cells or that area of tissue glowing a certain fluorescent tag color that you've given it. So we've done this in the rhesus macaque retina, and this is a cross-section of the retina, so if you took my eye out and then you sliced it like that, like so, and then you looked at it from the side. These are various layers of the retina that are necessary for visual processing when light comes into our eyes. And we stained it for amyloid beta, this A-beta molecule that we think might cause vision loss. So this large bump here is a drusen, and it's staining positively for amyloid beta, indicated by this red glowing color. And then these are photoreceptors in this layer of the eye, and they're also staining positively for amyloid beta. So we've used two antibodies, one with a green fluorescent label, one with a red fluorescent label, to detect the A-beta molecule that might cause vision loss. However, the keen eye will notice something very odd about this image. Now, I told you that antibodies are specific to the same molecule, so they should bind in the same locus of tissue because they're identifying the same thing. So if A-beta is in one section of tissue, both antibodies should bind to it there. So we should see this glowing in the same region of tissue. However, we're seeing it in mutually distinct layers of the retina. These antibodies aren't identifying A-beta in the same place. So this is a very odd pattern of expression. This isn't what we would expect. So one of two things has happened when you don't get a result that you expect in science. Either you effed something up, or you discovered something. So we wanted to shoot for the latter possibility. Figure out an alternative way of detecting how this molecule is made and how it might be involved in vision loss in this devastating form of blindness. So, as I previously told you, one molecule termed amyloid precursor mRNA, this messenger molecule, is necessary for making the A-beta protein. So it's a precursor in that way. And what if we could detect this precursor instead of the A-beta protein? Because we're seeing this weird thing happen with the staining of the A-beta protein. In fact, if we detect this precursor, then it will tell us where the cellular source of this blindness-causing molecule is, because proteins move around in tissue. So if we can detect this precursor molecule, that'll tell us where this stuff is coming from exactly. So I've created something called a riboprobe, an RNA detecting probe, that binds to this precursor molecule, APP mRNA, like Velcro in tissue. So you wash these riboprobes over retinal tissue, and then it binds like Velcro, and it has these fluorescent tags on it like antibodies do, so you can see where this precursor is being made and the cellular source of where the A-beta protein is coming from. So I'm going to go over a general conceptual overview of how I've made this, very briefly. So to begin, I have to select a region of the APP gene, the amyloid precursor gene, that I want to detect. And then I have to make a bunch of photocopies of that gene from a DNA sample that I've taken from a monkey. And then I have to synthesize my riboprobe from those little genetic photocopies using special enzymes. So I select a region of the APP gene using a computer database that I want to find and which cells express it. Then I have to make a bunch of photocopies of that gene segment, and then I use special enzymes, and I added fluorescent tags to make my riboprobe. So the end product that I get is a riboprobe that binds to mRNA in tissue like Velcro, and it has little fluorescent labels on it, so I can see where this stuff is made, because it'll glow. So there's various checks and balances I have to go through along the way as I'm doing this. First of all, I have to know that I've made the right photocopied gene segments of the APP gene that I'm interested in. So what I do is after I've made these photocopies of the gene segment, hopefully, I inject them into a gel. And this gel is a porous material. And I force these gene segments through the gel with an electrical current. Now, these gene segments are of a certain size. So smaller gene segments should force their way farther through the gel, and larger molecules will get stuck higher up in the gel as I'm pushing them through with this electrical current. So you'll get bands of gene segments that you've copied, hopefully, of particular sizes that are sorted in the gel according to their size. So on the left here, you can see the DNA ladder that I've injected into this gel. Now this has gene segments of known sizes. For instance, right here, this is a 400 nucleotide gene segment. So 400 A's, C's, G's, and T's in this little piece of DNA that I've injected here. Now this is my photocopied gene segment of the APP gene that I want to make. And it's supposed to be 373 nucleotides in length according to how I designed it. Now, if I've made the right product, then it should appear just beneath 400 base range, and it has. So I know that I've made my photocopied gene segment that I wanted to. So now I have to use some enzymes to synthesize my riboprobe, my end product. And I do the same thing after I've synthesized my riboprobe, hopefully. I check and make sure that I've actually done that using this porous gel material again. So I force it through the gel, and the gel sorts things by size, these gene segments by size. And on the left here, you can see a 500 base marker, 500 ACs, Gs, and Ts. On the right is my riboprobe, hopefully. I didn't know that before I injected it into the gel, so this is my confirmation that I've made the right thing. Now it's appearing just beneath the 500 nucleotide range. So I know, based on this band in the gel here, that I've made my final product, my riboprobe, the right thing that I was trying to make to begin with. So the last thing I have to do is actually rinse this riboprobe over tissue, retinal tissue that I was originally interested in, and try and find where APP mRNA, this precursor, the blindness-causing A beta molecule, is actually made. So where do I expect to find this stuff in the retina? Well, if you look at this pattern of A beta expression, the end product that I'm interested in, and we're trying to detect the precursor, well, there's a layer of cell that lie beneath or between these two layers of staining. So we're seeing A beta here and here. Now, we think that the APP, the precursor molecule to this protein, will be made in this cell layer called the RPE cell layer, the retinal pigment epithelial cell layer of the retina. Now, previous studies in rodents and in humans have shown that the RPE layer does often express APP, amyloid precursor. Now, we think it'll be expressed there, and it's also been highly implicated, these cells, in vision loss in AMD. So that's another reason to expect to find this disease-causing component in those cells in the retina. So why does this matter? Well, we're looking for a potential role of the A beta protein in AMD. We want to characterize it, know where it's made, and how it might cause blindness in people who suffer from this disease. In particular, we're looking for a cellular source of where the APP molecule, amyloid precursor, is made, and therefore where the A beta component is coming from. And finally, if we figure out where this stuff is coming from, that might lead to targeted treatments for people who suffer from this disease in the future, and that's the end goal that we're shooting for. So I want to thank Dr. Neil Melvin, Dr. Trevor McGill, Dr. Jonathan Stoddard, and Martha Neuringer, as well as Rich Wildman. Without these people, I wouldn't be standing here talking about this today. So, thank you.